Hello, welcome back to part 4 of our look at blood. And this diagram that is in front of you may look extremely scary as of right now, but over the next 10 minutes or so, I'm going to try to break this down in a step-by-step manner to go ahead and make each of these uh, stations, if you would, make sense. And so there are 13 steps that we are going to look at here. And really what we're dealing with is at the end of the 120 days of our red blood cell, what happens? What happens when this red blood cell officially dies and becomes broken down and recycled? There's some expensive parts to this cell that we need to take into account for. And then probably the most expensive part is the hemoglobin. We know that during the phase of a proerythrocyte or a reticulocyte, the proerythrocyte, during the period of a proerythrocyte, we know that hemoglobin is created and produced and added to the premature erythrocyte. Um, where does the material come from to be able to create and produce this hemoglobin? And the answer is we recycle the parts. And so what's happening during this whole process? And so let's go ahead and begin this process. Again, I'm going to break this down into 13 steps for our understanding, and then we're going to look at blood clotting as well. And so these I think go well together in one video for us. And so for red blood cell death and hemoglobin recycling, we're going to start right here with step 1. And step 1 is the red blood cell dies and a macrophage comes by and breaks it down. All right, and so it is broken down. Now, this breaking down of the red blood cells happens within the spleen primarily, and we'll talk about this um, later on when we get into the lymphatic system. Right, but the spleen is where the red blood cells go to become broken down. Also, the liver will do some breaking down of red blood cells, as the liver is the major detoxification center for the body. But the vast majority of red blood cell uh, breakdown through the use of macrophages is happening within the spleen. And so this is happening here in the spleen as well as the liver for the breakdown of our red blood cells. And what we end up with is we end up with two components, if you would, two segments of our red blood cells that begin to undergo um, recycling, if you would. And so that hemoglobin that is left over gets broken down into globin and a heme group. And so we're going to follow our hemoglobin as it's again broken down into the heme component and broken down into the globin component. And so let's follow our globulin. It's right here. Right? Globin is going to be broken down into amino acids, all right, and the macrophage is doing that. And so this big old cell that you're looking at right here, this pink thing, this is a macrophage. All right, it's broken down the red blood cells, it's taken the hemoglobin, it's divided it, it's broken it up into the heme group and the globin, and the globin is then re-broken down into its basic primary components, its amino acids. And the amino acids are simply released back into the surrounding tissue, the interstitial fluid, and as well as the blood for reuse and repurposing elsewhere within the body. So the globin gets broken down into the amino acids are released for protein synthesis elsewhere in the body. And we come back now. That was the easy part. Right? That was the easy part. Now we got to come back and we got to do something with this heme group. Right? And the heme is broken down again into two components, the iron component and the protein component. Right? And it's the iron component that we want to pay attention to here first. And so in step 4 of our diagram, iron is released from the heme, right? And that iron is inside of the macrophage, is transferred to a protein called transferrin. And it's the job of transferrin to safely transport iron through the bloodstream to the liver. So heme is broken down into iron and protein. Right? The iron is transferred to a protein called transferrin, whose job is to safely transport the iron molecule through the bloodstream to the liver. Right? To the liver. Right? Now, from here, the liver can store the iron if iron is in excess, or the liver can take the iron that is on the transferrin. Right? It can move it to ferritin. Right? Ferritin is what is actually going to store the iron. All right. And when needed, ferritin can pass the iron back off to transferrin. Transferrin, again, will safely transport the iron through the bloodstream and send it to the bone marrow. Now, why is this important? Why is this critical? Well, because we know that it is within the red bone marrow where red blood cells are being produced. This is where the proerythroblast is going to be located, which means it's within the red bone marrow where hemoglobin is going to be added to that proerythroblast. And so the iron needs to be moved back to the bone marrow, the red bone marrow, to be converted and transformed back into the hemoglobin. And so once again, let's follow this pathway. Heme gets broken down into the iron within the macrophage. The iron is transferred to a protein called transferrin, whose job is to transport it through the bloodstream to the liver, where transferrin passes the iron off to another protein called ferritin. Ferritin's job is to store excess iron within the liver. When needed, ferritin can pass the iron off to transferrin once again, which will safely transport the iron back to the red bone marrow in the bone to go ahead and be converted into hemoglobin. Here's the catch. That production of hemoglobin is dependent upon vitamin B12. It's dependent upon vitamin B12. And we talked about that. We talked about that in relation to, or at least in the lab component, we talked about that. And so we know that vitamin B12 deficiency impairs the ability for iron to go ahead and be converted into hemoglobin, which creates anemia. Right? That is one of the leading causes of anemia. And so vitamin B12, we know we absorb through the small intestines and is directly dependent upon intrinsic factor. And so if the stomach is not secreting intrinsic factor, vitamin B12 will not be absorbed, which means you will have a deficiency in the ability to produce hemoglobin. And that will lead to an iron deficiency, or that individual being anemic. All right. Specifically pernicious. All right. Now, of course, we know that the iron mixing with the vitamin B12 to help create the hemoglobin with the globin that has now been produced under the direction of erythropoietin is going to create new red blood cells that'll circulate for the 120 days and come back to the spleen or the liver or the bone marrow, where it will be broken down by the macrophages. All right. So that's the journey of our iron, but we still have the other part of this heme group, this other protein aspect of the heme group that we need to consider. And so what happens over here in step 9 is that this protein base, this non-iron protein base portion of the heme group, is converted into what we call biliverdin, and biliverdin is transformed into bilirubin, and that bilirubin is stored within, once again, the liver. All right, now, bilirubin is free to circulate through the bloodstream, doesn't need a transport protein or anything like that. And so the bilirubin created within the macrophage is released into the bloodstream and travels to the liver where it is stored. And that bilirubin will become part of bile, and we'll talk more about that when we get into the digestive system. Now, that bile is going to be secreted from the liver. It's going to be concentrated in the gallbladder. It's going to go down the common bile duct into the pancreas, where it's going to join the hepatopancreatic duct. And eventually, this bilirubin inside of the bile will be secreted into the small intestines. From the small intestines, it'll go into the colon or the large intestines, where that bilirubin will be converted into urobilinogen. And it is the urobilinogen that gives feces that its characteristic brown color. And that conversion of bilirubin into urobilinogen is directly under the regulation and control of bacteria living within the large intestines. And then it is secreted through the feces, or some of that urobilinogen will actually be reabsorbed into the bloodstream. It'll be passed off into the kidneys, where it'll be transformed into urobilin and then secreted through the urine. And so that bilirubin has two different pathways for being completely broken down and gotten rid of, either as urobilinogen or as urobilin, whether it's being excreted through the feces or being secreted through the urine. And so I would suggest maybe pausing the video here and kind of rewinding and maybe going back through that, and make sure that you've got this broken down step by step for your understanding. And there's nothing wrong with pausing the video now and then rewinding and rewatching this first part, because as you can guess, there's probably a series of questions coming up right after I click to the next slide. And sure enough, you just got done answering some questions, as I promised. And so hopefully that was a good little review of that process. What I want to spend the next 10 minutes or so on is blood clotting. And so let's go through and let's look at what is the homeostatic mechanisms here that regulates and controls the clotting of blood. In other words, you've got some kind of damage that is happening within the body. How do you mitigate blood loss? And again, this is a homeostatic mechanism. All right. And so we have two different pathways. We have two different instances by which we can go ahead and initiate the clotting of blood. We have an extrinsic pathway and we have an intrinsic pathway. Extrinsic is going to happen within the tissue. It's going to happen within the tissue, right, outside of the capillary beds. And so you've got damage to some tissue area, and maybe along those same lines, you've damaged some capillaries in the area. Right? And you need to now go ahead and reduce the amount of blood loss or bruising that is happening underneath of the skin. Your intrinsic pathway happens within the actual capillary or arterial or vein itself. And so this would be damage to the endothelial lining that is lining the lumen of that blood vessel. And so we've got two different pathways here that we need to consider. Both pathways, though, lead to the same end result. All right? And so here's your extrinsic pathway right here. All right? Here's your intrinsic pathway right here. Both of them lead to the production of prothrombinase. And prothrombinase is an enzyme that is key in what we call the common pathway. And it's within the common pathway where we see the production of fibrin. In other words, it's within the common pathway where fibrinogen is reconstituted as fibrin to actually create the blood clot. So we have two different scenarios in which blood clotting can be initiated under. The extrinsic pathway is a little bit faster in response, mainly because of the tissue damage that is initiated. And so um, uh, the damaged tissue, the cells within the layer of the tissue that has become damaged, begins to release something called tissue factor, TF. And the presence of tissue factor, when combined with calcium ions, so calcium ions here acts as a regulator. Calcium binds to the protein tissue factor, and what that does is it allows that tissue factor to then activate factor X. So there's 13 different clotting factors within the body. Factor X is one of those clotting factors. All right, and that is key in the production of prothrombinase. So you have to activate factor X. Well, how do you activate factor X? By having calcium bind to tissue factor that has been released from the damaged cells within the area. Factor X will go ahead and join with factor 5. And once again, what allows factor X and factor 5 to merge is the presence of calcium. And so prothrombinase is the merging of both factor X and factor 5 in the presence of calcium. Right? That's the extrinsic pathway. Within the intrinsic pathway, we see something a little bit different. It's a little bit slower to react because usually the leaking is not as severe, or the blood loss is not as severe, and so we don't have tissue factor that is present. Instead, what ends up happening here is you begin to leak platelets, and through the damaged endothelial cells. And when you start to leak those platelets, factor 7 becomes activated. Factor 7 merges, or I should say calcium binds to factor 7. And what that leads to is the formation of factor X. All right, and then factor X once again joins with factor 5 in the presence of calcium to create to create prothrombinase. And prothrombinase can then go ahead and be introduced into the common pathway. And it's within the common pathway where we once again see calcium having to bind to prothrombinase. Right? And this time what ends up happening here is when calcium binds to prothrombinase, we cleave it, we shorten down that protein, and we create prothrombin. Right? We create prothrombin. Prothrombin is then converted into thrombin. And thrombin then again binds with calcium. Calcium is the key regulator here. Thrombin, in the presence of calcium, can then go ahead and drive the formation of fibrinogen into fibrin and activates the presence of factor 8. Why is factor 8 important? Because factor 8 stabilizes fibrin. Remember, This is a positive feedback mechanism. This is not negative in any way, and it's not natural for fibrin to be present. So the body tries to work against the formation of fibrin because of clotting. If you have fibrin forming within the vessels, you're going to have a blood clot. And so we try to not form the fibrin. And so fibrin is highly unstable. It wants to become fibrinogen. That's its happy state. Right? That's where it's most comfortable. And so thrombin in the presence of calcium forces fibrinogen to become fibrin once again. But thrombin also has to promote the release in the presence of factor 8. Factor 8 binds to fibrin and prevents it from becoming destabilized. And so the presence of fibrin then goes ahead and aids in the formation of the blood clot. Understand, this is a very localized paracrine pathway. So this is not happening throughout the body. This is only happening within that damaged area or that damaged zone. And again, the purpose of this is to mitigate any further blood loss. Um, this is a quick overview. This does get a little bit more complicated. Again, we've only touched on a few of the clotting factors that are really involved with this whole process. But the key ones are factor X, factor 10, factor V or factor 5. And the reason why those two are important is because they're the two clotting factors in the presence of calcium that is going to create prothrombinase. Prothrombinase enters the common pathway and is converted into prothrombin, then into thrombin into the presence of calcium. Thrombin, again in the presence of calcium, then drives fibrinogen to become fibrin, secretes or activates factor 8, and factor 8 binds to fibrin, preventing it from going back to becoming fibrinogen. So it stabilizes the fibrin to allow the blood clot to go ahead and form. And so once again, I would suggest maybe pausing the video, rewinding back to about the 13-minute marker, and maybe rewatching this, making sure that your notes are good, making sure that you've got a good understanding of this, because you know when I click to the next slide exactly what is going to happen, and that is a set of questions is going to come up. And as I warned, a set of questions did come up. Uh, and so the last little thing that I want to cover here is kind of a review of things that we've already talked about. So I'm not going to spend too much time on this, but white blood cells. We've spent a lot of time talking about erythrocytes, so let's give a little love to the leukocytes. Um, Leukocytes or white blood cells we know contain a nucleus. That is evident from what we have seen in lab. No hemoglobin is present because it's not there to carry out oxygen transport. Instead, we know that the leukocytes carry out immune response. And we know that we have two types of leukocytes that we need to be concerned with. Granulocytes and agranulocytes. Right? Granulocytes or granular leukocytes have little tiny protein granules that are all throughout their cytoplasm. And what distinguishes each of these types is really the shape of their nucleus. The nucleus is that darkly stained purple structure that is in the middle there. And so we have three types of granulocytes that you need to be able to differentiate and identify. The neutrophils, the eosinophils, and the basophils, what I like to refer to as the three Phil brothers. All right. Neutrophils, we already know, are the first responders. We know that they can engage in phagocytosis, so they really act as a macrophage. We also know that they can engage in paracrine and autocrine function, through the release of cytokines. We know that they're about 60 to 70% of the overall white blood cell count or percentage. Eosinophils. Oh, by the way, we know that neutrophils are characterized by having 3 to 5 lobes. Right, so that nucleus is broken down into very distinct 3 to 5 lobed areas. Um, Eosinophils, we know, account for 2 to 4% of the overall leukocyte percentages. We know that they are activated mainly during allergy responses and parasitic infections, and their nuclei, I like to say, looks like a pair of shoes that have been like tied together and thrown over an electrical line, or maybe a pair of boxing gloves. And so you get these 2 lobes, and then a tiny little membrane section of that membrane, right, will then join the other lobe together. Whereas with neutrophils, you get 1 lobe, 2 lobe, 3 lobe, 3 to 5 lobes. One, two, 3, 4, and 5. All right. Whereas eosinophils, 2 distinct lobes, and those lobes are connected with a thin piece of nuclear membrane. All right. Basophils, we know that basophils are about 1%, if not less. We know that they secrete heparin and histamine, and heparin is a blood thinner. Histamine is a vasodilator, but a bronchial constrictor. Right? And typically, the nuclei, first of all, you don't usually get to see too much of the lobe, 1, 2 lobes that are connected by a thicker region. But these have a lot of those protein granules that are in there. Very, very heavily pixelated or granularized are the basophils, and that's because those granules are secreting things like heparin and histamine, right? Which ironically, we take antihistamines to block the release of histamines that are there to go ahead and cause you to help remove the allergen from your bodies. But they're the 3 Phil brothers. Right? And then we have the agranulocytes, and that means these are leukocytes that lack the granulation within the nucleus, and or I should say, within their cells, within the cytoplasm, and the nucleus is much, much larger. And so think about the lymphocytes, which account for 20 to 30% of the overall um, structure, right, or a percentage of the white blood cells. And again, we see lymphocyte numbers go up in the presence of cancer, viral infections, bacterial infections, or just when called upon, but usually by the nucleus, right? Or by the sorry, not the nucleus, by the neutrophils, and their nuclei tend to be very large. Right? They stain nicely purple, and they take up almost the entire cell, such as that. All right. And then your monocytes are really macrophages. They aid the neutrophils in cleaning up debris and helping to kill uh, pathogens that might be within the cell. And so about 3 to 8% of the overall white blood cells that we run across are monocytes. And the nuclei of the monocytes tend to be more kidney bean shaped, right, or neck pillow shape, like that. Right? Again, taking up a large component of the cell, right, just like that. And just to tie this all together, just so you can kind of see what each of these types of leukocytes look like, this diagram here is nicely laid out. You can see the very clear lobes in the neutrophils. You can see the differences are between the eosinophil and the basophil, although the eosinophil is a little sketchy. The basophil is a nice basophil. And you've got your monocytes with your like neck pillow or kidney bean shape, and then your 2 lymphocytes, your B cells, and your B cells and your T cells. You will not have to differentiate between the two, so don't necessarily worry about that. Um, but this gives you a good overview of the types of leukocytes, the granulocytes, and the agranulocytes that you will encounter. And so I would suggest that you go ahead and pause the video because you know you're going to have some questions coming up, and kind of review this, jot down any questions you may have. Keep studying. I know it's a lot of material, but you guys are doing great. As always, let me know if you have any questions, and I'll catch you on the flip side.